welcome back. I hope you had enough food to at least survive another hour of good science. I didn't have to say anything else, right? Like there's no urgent messages or anything. There are still people coming in. Well, it's good that you all came back. At least 75%, I think. So that's good. But yes, so we're going to move to the last lecture of today by Dr. Aude PANATIER. Hey, that rhymes. She's from Bordeaux. She was born there. Yes. And she's a very good scientist. Like she is a very instrumental person in the field of astrocytes. So of course we all know. Well, this is kind of a glia-loving crowd, I think. That you know. There's like all these brain cells, and most of them, most people, they study neurons, but there's also glia, and you know, Aude has been very influential in showing that astrocytes are very important for regulating behavior, and specifically she studied how astrocytes, you know, regulate synaptic plasticity. She used to do this in the hippocampus in the hypothalamus with oxytocin, which is my favorite neuropeptide. But now she's working in the hippocampus, and yes, I would welcome her here. She's gonna talk about D-serine, so glial transmission, and how neurons and astrocytes talk to make you behave and remember things.
Thank you very much. Do you hear me? It's okay like that? Okay. Thank you, Aaron. Thank you, Monse. Thank you, all the organizing committee and scientific committee. It's really nice to be there in a really good atmosphere. Thank you so much. It's really cool. So, yes, today I will talk about astrocytes, and I will, in fact, give me this journey and a bit my journey too, you will understand. So in the brain, as you all know, information is transmitted from one neuron to another one at the level of the synapse. But in fact, only half of the brain is composed of neurons, the human brain. And the other half is composed of glial cells. So my favorite glial cell, since more than 25 years now, is the astrocyte that is well positioned in between blood vessel and synapses. Due to this key position, very early at the end of the 20th century, it has been proposed that astrocytes can pick up the glucose from the blood, transform it into different metabolites like lactate, and therefore then give lactate to the neuron as an energetic source. At the same time, it has been proposed that because astrocyte is also present at the level of the synapse, astrocyte is able to hear from what is happening at the level of the synapse, and in turn to answer, to participate to the regulation of the neuronal communication, of synaptic transmission. So today, if I have to present the tripartite synapse, it's like that. I hope and I think that in a few years it will change, but for now it's the way I see it. So you have the pre and the post-synaptic element, the associative one here. And so during synaptic transmission, once the action potential arrives, this is inducing at the level of the glutamatergic synapse the release of glutamate, and glutamate will activate not only post-synaptic receptor, but also receptor present at the level of the associative process. This will lead to an increase of calcium in the astrocyte, and in turn, the astrocyte will release gliotransmitters. So several gliotransmitters have been shown until now. So the classical one, glutamate, D-serine, ATP, and more recently, GABA. So I will not really talk today about ATP, but I will just mention the fact that when I was in postdoc in Montreal in the lab of Richard Robitaille, we have shown that contrary to what was thought at that time, astrocytes are able to detect the glutamate that is released after the arrival of a single action potential. Because in fact, if you go directly to the synapse, you can see that the astrocyte is able to detect a single vesicle of neurotransmitters, so glutamate released, through mGluR5, and this is leading to local calcium events that will never propagate. It will stay in the compartment close to the two neuronal synaptic elements. And in turn, the astrocyte is releasing purine that will be ATP that is degraded into adenosine and that will activate presynaptic A to A receptor. And this will lead to an increase of the efficacy of synaptic transmission. This was shown in juvenile animals, and more recently we have shown that this is also true in adult mice. mGlu5 is doing the same job in adults. And it's a bit more complex because this only occurs if some sparse synapses are activated at the same time. As soon as you activate with only a single but several synapses at the same time, this process stops. What does that mean? This is what we are investigating. So the other glial transmitter I'm working on since my PhD is D-serine. So what is D-serine? So D-serine is a coagonist of glutamatergic NMD receptor. As you know, to be activated, this receptor needs the binding, the concomitant binding of two ligands. The coagonist on the glycine binding site, that can be either glycine or D-serine, on the gluon1 subunits, and the glutamate on the glutamate binding sites on the gluon2 subunits. In addition, to have a functional receptor, the magnesium needs to be released, and this is arrive during depolarization. So, all right. What did we do? When I was in PhD, we have shown that in the supraoptic nucleus, in the hypothalamus, astrocytes, by releasing D-serine, control the activity of NMD receptor, because you need, again, the binding of both the coagonist and glutamates, control the activity of NMD receptor, and therefore the induction of LTP and LTD. Surprisingly, if you do exactly the same experiment, but this time on acute brain slices from lactating rats, what happens? It happens first that the associative processes are not really present at the level of the synapse. They are retracted. And therefore, the availability of D-serine in the synaptic cleft is impaired. Because of that, the number of NMD receptors that will be available for activation will be decreased. And therefore, the classical protocol that normally is inducing LTD does not at all modify the efficacy of synaptic transmission. Pairing protocol, so moderate protocol that normally is involving to induce LTP, this time is inducing LTD. And in fact, to induce LTP in the supraoptic nucleus, when the associative processes are retracted, you need to use high-frequency activity, like the one that is involved in the release of oxytocin in the blood to induce the milk ejection at the level of the mammary gland, and therefore to have this reflex, because this will induce then the activation of presynaptic glutamatergic synapses to activate in a firing mode the magnocellular neuron. And in this condition, you can induce LTP. So then I left to Montreal, and I started to study the role of astrocyte in the hippocampus, the story I told you just 2 minutes ago, regarding purine. And at the same time, in the lab, in Stéphane Olier's lab, they decided to look whether D-serine could also be the coagonist, the synaptic coagonist, the coagonist of synaptic NMD receptor in the hippocampus, a structure in which we know that LTP is playing a key role in this structure. So what they have shown, like in the hypothalamus, they show that D-serine is the coagonist of synaptic NMD receptor. What they have shown, in addition, is that for this release, calcium is required. So the release of D-serine is calcium dependent. Then, a little bit later, they show that, okay, astrocyte release D-serine, and D-serine is a coagonist of synaptic NMD receptor, but this is not the case at the extrasynaptic site. For extrasynaptic NMD receptor, the coagonist is not D-serine, it's glycine. And then I came back in Bordeaux, and I get a position, and we continue to work on D-serine to better investigate how this process is working. And Mark and Misa show that the calcium that is involved in the activation of NMD receptor during the LTP induction is coming from reticulum endoplasmic and require the activation of IP3 receptors. Then we have shown, so not me, but in collaboration with Giovanni, Valentin performed the electrophysiological experiment in the team, and 2 other students in the lab of Giovanni performed behavioral experiment as well as in vivo electrophysiology. And they show that the activation of CB1 receptor is required to control the availability of glycerin in the synaptic cleft, and therefore the activation of NMDA receptor, the induction of LTP, and memory. More recently, we have shown that glucose, in fact, we are not the only one, but we know for now that glucose, glycerin is coming from glucose, so once glucose is uptaken by the neuron, glucose can be transformed in lactate, but also in L-serine, which is a precursor of glycerin. And more recently, with Giovanni, we have shown that, in fact, this pathway producing lactate is playing a key role in promoting L-serine. In fact, once a CB1 receptor is involved in the synthesis of lactate, and once lactate is synthesized and released, the lactate will activate associative HCAR1 receptor that will promote the L-serine pathway, and therefore the D-serine production. So now there are still a lot of questions, and one of the questions is why? Why D-serine in the synapse and glycine extrasynaptically? One possibility could be the fact that what is the difference is that at the synapse, the associative process is there. And remember, in the hypothalamus, we have shown that when the associative processes are retracted, the level of D-serine in the synapse is impaired. So we thought, all right, perhaps some cell adhesion molecule, some CAM, like the F receptor, so could be involved. So what is a F receptor? So here I'm talking about specifically, but it's the same for all the F receptor. You have here a receptor, so it's the FB3 receptor, and the particularity of those receptors is that the ligand is not a transmitter. It's another transmembrane protein. So here you have a transmembrane protein that, for example, can be expressed by the astrocyte, and here you have the ligand binding. And its ligand, it's another transmembrane protein that is expressed by another cell. So you need that both membrane, the associative one and the, let's say, postsynaptic one, are close together to being able to activate the receptor with the ligand. Is it okay? Okay. So why this receptor? Because in fact, in pure associative culture, it has been shown that if you add the ligand of the receptor, so ephrin B3, the ligand, this will induce an increase of D-serine release. And these do not occur when the astrocytes do not express the receptor. In addition, in a wall knockout for FB3 and FA4 receptor, it has been shown that the LTP was impaired. Therefore, Valentin, when he was in PhD, and Sarah, when she arrived in the lab as a postdoc, they wanted to better understand whether associative FB3 receptor controls synaptic NMDA receptor function. First, the NMDA receptor activity, the induction of long-term synaptic plasticity, and thanks to our key collaboration with Giovanni Marsicano, memory. So how did they do that? So they perform experiments on, let's say, adult mice, and they perform acute brain slices. So the idea is to measure the activity of NMDA receptor. And for that, we used extracellular field recording. So you have your acute slice, you position your pipette outside the cell, and you stimulate. So once you stimulate, this is inducing the release of glutamate, and glutamate will activate postsynaptic receptor, and the first one is the well-known AMPA receptor. Once the receptor is activated, positive charge will enter, and outside what you see is an increase of negative charge, and therefore a decrease of the potential that you are recording extracellularly. But if you do that classically, you will not be able to record the activity of an NMDA receptor because, as I said, at the resting membrane potential, the receptor is blocked by magnesium. So to study the extracellular field NMDA receptor, what we did, we used pharmacology. So first, for sure, you block the GABA transmission, and then you block the AMPA receptor with NBQX, and you remove, or let's say decrease a lot, the magnesium ions in the ACSF. And therefore, like that, you can record the extracellular field NMDA receptor. So this is what we did. Then, it's not a classical receptor. So this receptor is a ligand transmembrane protein. So its ligand is another transmembrane protein. How do we do to activate it? So first of all, more the receptor is clustered, higher the activity is, less the receptor is oligomerized or clustered, less the activity of the receptor is. So what we can do is to use chimera. So what you do, you use some chimera with the ephrin B3, so the ligand part, that you can cluster using anti-IgG. This is your clustered ligand to activate the receptor. But it's a big component, so you need to control for that. And therefore, we have the control clustered non-ligand, so the control IgG. Exactly the same thing. You have anti-IgG that will cluster the human IgG1, but this time there is no ephrin B3 part. So this cannot activate the ephrin B3 receptor. And as I said, to decrease the activity of the receptor, we need to decrease its level of clusterization. Therefore, we do exactly the same things, but this time without an antibody, anti-IgG1. So, okay, first question. Which concentration of the chimera, let's say the ephrin B3, do we have to use to activate, to increase the activity of an NMD receptor? So what they did, they used different concentration of the clustered chimera, and they measure the activity of the NMD receptor in function of the concentration. And therefore, once you start to increase the concentration, this is inducing an increase of the NMD receptor activity. And we decided to use the concentration, the minimal concentration that is inducing the higher effect. Now the question is, all right, you activate the FB3 receptor using the ephrin B3 ligand, and this is inducing an activity of the NMD receptor. But we wanted to know whether this could induce an increase of dicerin and therefore an increase of NMD receptor activity. So to answer this, specifically, we needed to study the coagonist binding site occupancy, so the occupancy of these sites specifically. Exactly the same technique. Extracellular field NMD receptor, you stimulate, this is inducing the release of glutamate and dicerin, and you can record a field NMD receptor. Now, classically, the coagonist binding site is not totally saturated. So if you add exogenous dicerin in the perfusion bath, this will induce a small increase of the extracellular field EPSP of around 20%. So it's close to saturation, but it's not saturated. And like that, you can study the coagonist binding site occupancy. And it's exactly what we did then. So first, we record the activity of an NMD receptor, and we apply the control chimera. And when you do that, you continue to study the activity of an NMD receptor, and the control chimera does nothing. Then, in the bath, you add dicerin, and as I said, the application of exogenous dicerin is inducing around 20% of increase of the activity of the NMD receptor. Now, what happened with the ephrin B3 ligand? When we do exactly the same thing, but this time we add the ephrin B3, this is inducing an increase of the activity of an NMD receptor of around 20%. And in addition, the addition of dicerin does not have really an effect, and the effect was occluded by the presence of the ephrin B3 ligand. But you can tell me, all right, why not? But perhaps this can induce something else on the receptor. So let's do the experiment in another way around. This time, we apply dicerin from the beginning. We saturate the site. And if the effect of the ligand is only due to an increase of the dicerin, we should not see additional effect. But if the ephrin B3 does something else, we should see it. And in fact, the application of ephrin B3 in the presence of dicerin did not have any additional effect, showing that the activation of the FB3 receptor is inducing an increase of the activity of an NMD receptor through an increase of the availability of dicerin. But then, this receptor is expressed not only in astrocyte, but also in neuron, and it's the same thing for the ligand. So we needed to know whether astrocytes were involved in. So for that, we this time record the extracellular field NMD receptor directly through the astrocytes. So the astrocyte has a particularity. Its input resistance is really, really low. So because of that, so it's a mess, because when you do electrophy, the astrocyte at the end is boring because the input resistance is really low. You cannot really record currents. But anyway, but it's also good because of that, you can record the extracellular field occurring at the level of the synapse that are in close opposition with the astrocyte you are patching. And due to that, you can introduce whatever you want in the astrocyte. And this time, what we did, so this is a classical astrocytic field NMD receptor. And what we did, we introduced in the patch pipette a calcium clamp solution. So here it's a patch experiment. You record the extracellular field NMD receptor, so the neuronal one, using a control intracellular solution. What we do, so this is the extracellular field, you apply the ephrin B3, and this is inducing the classical 20%. Everything is going well. Now, when we block calcium activity in the astrocyte, remember, the release of this ephrin is calcium dependent. When we block the calcium, we clamp the calcium specifically in astrocytes, ephrin B3 activation did not have additional effect, meaning that the increase of this ephrin induced by the activation of FB3 receptor is going through the astrocytes. So, all right. Now, let's say, you will see I have additional experiment after, but let's say associatic FB3 receptor is involved. I cannot say that. See, if you activate the associatic FB3 receptor, this will lead to an increase of this ephrin. Now, the question is whether this is really true. So, does endogenously astrocytes through FB3 receptor control the activity of NMD receptor? So for that, we need to inhibit the receptor. And by doing so, if you inhibit the receptor and you see an effect on the activity of an NMD receptor, this means that, yes, endogenously, FB3 receptors are involved in the control of the availability of synaptic NMD receptor. So again, we did the same experiment, but this time using unclustered IgG. So this is a control experiment, activity of an NMD receptor during time. You apply the control, nothing, you apply diserin. This is inducing an increase of the activity of an NMD receptor. Then when we block, we decrease the activity of those receptors using unclustered ephrin B3. This is inducing a decrease of the activity of an NMD receptor that is rescued by the exogenous application of diserin, meaning that, yes, associatic FB3 receptor control the availability of synaptic diserin and therefore the activity of NMD receptor. So then we ask the question whether this receptor, by controlling diserin, regulates also the induction of LTP, so long-term synaptic potentiation. So the same experiment, this time it's classical extracellular field EPSP. So here this field is not due to NMDA, it's due to the activation of AMPA receptor. So you record the activity of the AMPA receptor so extracellularly, so the classical field EPSP, during a baseline period, and then you induce LTP. We induce LTP using the classical HFS, so 100 Hertz, 1 second, 3 times, 20 seconds apart. And when you do that, the application of this protocol is inducing the classical persistent increase of the synaptic strength. Then what happened when we block the FB3 receptor? So we use the unclustered FB3 ligand, and by doing so, this was inducing an impairment of the induction of LTP. Therefore, we could rescue that using diserin, meaning that this impairment was really due to a decrease of synaptic diserin. So then I remember Luigi, he was in the lab of, he is in the lab of Giovanni Marticano, and at that time he told me, yeah, you know, we should develop something to being able to go more in vivo, to manipulate the receptor. So what Luigi did, he developed one SH strategy to decrease the expression of FB3 receptors, specifically in astrocytes. So using 2 AAV, 1 control, allowing the expression of GFP. So now we have several ones. This one was a red virus allowing the expression of the SH-FB3. We have now the green version, and we have also the control version with the scrambled shRNA. And what we did, we injected either the control virus or the SH-FB3 virus in 7-week-old animals. We waited 5 weeks, and then first we perform again LTP experiment on acute lysis. And therefore, when the receptor is specifically knocked down in astrocytes, the LTP is impaired in adult mice. And this is rescued by exogenous decellin. So it's really due to an impairment of decellin. And then we asked whether this could have an impact on memory. So Luigi performed that time a novel object recognition memory that you, I'm sure, all know, and potentially better than me. So he used the classical L-maze. So he put first the mice during 9 minutes for the habituation phase. The day after, he put 2 objects that were the same, exactly the same, in both arms. And then the day after, it was the test day, he replaced 1 object by a new one. And the classical behavior of the mice is that he will pass more often time close to the new object in comparison to the other one. So first of all, the SH didn't have any effects on the exploration time. Then we looked whether it has an impact on the memory performances, and as you can see, the inhibition of the expression of the FB3 receptor is impairing the memory performance of the mice. And then, in fact, just after the acquisition phase, he injected IP the decellin, and we look at the performance the day after. While the injection of decellin didn't have any additional effects in control mice, it rescued the behavior in knockdown FB3 mice. Meaning that, yes, astrocytes, by expressing FB3 receptor, control not only the activity of NMD receptor and the LTP induction, but also memory performance. So, regarding this work, I wanted to put the timer, but I did not. It's okay. I have time. Okay. So there are still some questions. So is the activation of this receptor involved in an increase of calcium? Because remember, CB1 is there, and CB1 is doing exactly the same stuff as the FB3. You remove one or the other, you have the same impairments. Interestingly, it has been shown that sometimes the F receptor can be linked to calcium. So perhaps this one is. What is interesting too is that it has been shown that the receptor is also linked to PIK1 and PKC alpha, so controlling potentially decellin synthesis. This has been shown only in culture. And as I just mentioned, interestingly, if you impair this receptor, you have exactly the same impairment that if you impair the CB1 receptor. How do they interact? This is what we will do. We are studying now. There is another question. So calcium, from where is coming calcium? So I told you in the introduction that we have shown in 2017 that if you impair IP3 receptor, I will show you, specifically in astrocytes, there is no LTP. But surprisingly, a bit before, it was shown that in a knockout for IP3 type 2 receptors, the LTP is still preserved. And IP3 type 2 receptor is believed to be the IP3 receptor expressed specifically in astrocytes. So we were a bit, you will see, we were a bit surprised regarding the previous data, because so what Mark and Misa did, so Mark performed the electrophysiological experiment and Misa the calcium experiment. what Mark did, he introduced specifically into the neuron, so again, it's a patch of neuron, and he recorded the field EPSP through the astrocyte. And he introduced specifically in the astrocyte heparin, which is a pan inhibitor of IP3 receptor. So if you inhibit all the IP3 receptors specifically in the patch astrocytes, what happens is that HFS is not inducing the classical increase of calcium that normally occur in controlled conditions like here. Now what happens regarding LTP? So regarding LTP, if all the IP3 receptors are inhibited, the LTP is also impaired. And what did I do here? If you saw all my presentation, if you have to guess the experiment I did here, it was, I think I heard it. I add exogenous glycerin to show that this impairment was really due to a decrease of glycerin in the synaptic cleft and therefore decrease of NMD receptor activity and LTP induction. And this impairment was rescued by glycerin. But again, I told you, at that time, it was known that in a specific IP3 type 2 receptor, the LTP induction is not impaired, the glial transmission is not impaired. So it was a bit strange. And we reproduce exactly the experiment as it was before. So it's total knockout of IP3 type 2 and IP3 type 2/3. So this is classical field EPSP. And as before, we could reproduce exactly the same thing. The LTP is not impaired in IP3 type 2 cow. So we thought, all right, perhaps IP3 type 2 are not the only IP3 receptor expressed by astrocytes. So the classical idea is that neurons are expressing IP3 type 1 and astrocytes are expressing IP3 type 2 receptors. But we were a bit surprised by the electrophysiological experiment, so therefore we wanted to go further. And we asked whether perhaps astrocytes are expressing also IP3 type 1 receptor, like neurons. So this is a classical immunohistochemistry for IP3 receptors. So you can see that it's expressed everywhere in the hippocampus. So we are interested in CA3-CA1 synapses. And here, the neuron express IP3 type 1 receptor as expected. Then, if we use specific, so here it's a GFP labeling of the astrocyte, but the membrane one. So we see the GFP at the level of the membrane of the astrocyte, and we can also recognize the astrocytes thanks to the GFP labeling. And now if we look at IP3 receptors specifically in astrocytic process, expressing again the GFP, at the level of the membrane, can we see IP3 receptor type 1 inside the astrocyte? And in fact, the answer is yes. Here you can see IP3 receptor type 1 specifically in astrocytic process along GFAP, close to GFAP, inside, but not only because here there is no GFAP, so also in the small astrocytic process, inside the astrocytic process. So yes, apparently, astrocytes also can express IP3 type 1 receptor. So now we wanted to go further and investigate whether this receptor, by knocking it down specifically in astrocyte, has an impact on LTP induction. So again, exactly the same experiment. So this time on IP3 type 1, either control or knockout mice, adults. So this one was a control mice, so classical control AAV, GFAP, GFP virus. And Mark performed the classical experiment of LTP with or without glycerin. He could induce LTP. Now, what happened in IP3 type 1 knockout mice, specifically in astrocytes using another AAV virus? So the same thing, we inject the animal at 7 weeks old, we wait 5 weeks, and we perform experiment. And this time, the LTP was really impaired and rescued by the exogenous application of glycerin, meaning that, yes, as a neuron, in fact, astrocytes express IP3 type 1 receptor, and those receptors are involved in the release of glycerin that is required for the induction of LTP. We use another strategy thanks to Mark, because Mark and Misa are coming from the lab of Miko Shiba, with the guy that cloned those receptors and developed a lot of tools. And they developed a specific tool, which is an antibody that will specifically block the function of IP3 receptor. So this time, exactly the same experiment, this time by patching the astrocyte, Mark could introduce in the astrocyte either a control IP3 receptor type 1 antibody that does not block the function. It recognizes the receptor, but it does not block the calcium. And another antibody that this time is close to the channel and that will close or impair and block the release of calcium from the internal store. So this is exactly what happened. When you block the IP3 receptor type 1 function, specifically in astrocyte, the LTP was impaired and rescued by the application of diserine, an additional proof that IP3 receptor type 1 are the one involved in diserine availability. So to conclude this story, in fact, I didn't show everything, but what I show you today is that IP3 type 1 are also expressed in astrocytes, and they are the one involved, as I mentioned several times now, in the availability of diserine, NMD receptor activity, and LTP. And we didn't test memory, but my guess is memory. So I've done, just to conclude what I show you today, we have shown that once uptake, the glucose can be synthesized either in lactate or a lot of other stuff, but let's say in lactate or L-serine. Lactate will promote L-serine production, which is required for the activity of NMD receptor, LTP induction, and memory. Two receptors for now are key players in the availability of those synaptic diserine. Those two receptors are CB1 and HB3 receptors. And all this process depends on calcium. And regarding the diserine that is necessary for LTP induction, it requires the activation of IP3 receptor type 1. So now the question is whether those receptors, the CB1 and diserine, how are they connected? And are they also playing a key role in glycine availability that is now necessary for the activation of extrasynaptic receptor? We still don't know whether glycine is coming from astrocytes and whether this process is also involved in the presence of glycine at extrasynaptic sites. So here you have all the persons that contributed the past member of the lab. So I mentioned all of them during the presentation. This is due to key collaboration in Bordeaux, as well as in Paris and in Japan, and thanks to the nice platform we have in Magendie, as well as in the department. Thank you so much also for your attention. Maite.
No, we're coming. Oh, that's so sad.
Yeah, I think they will activate.
Okay, shouting.
Thank you.
So my question is, the IP3 are two receptors now optimized. They have a dramatic decrease of calcium activity in those astrocytes. What about the non-alkylated one? Does it also affect the calcium activity in those astrocytes somehow?
I don't have the answer, but so For sure, when you block all the IP3, it decreases. For the IP3 type 1, it's a bit more complex. So you need to go to the synapse to really see an effect, because those ones are involved in the fast calcium events that potentially cannot really propagate. The one you see with the IP3 type 2 is a calcium that is propagating in the process. And so it's a good question. My guess is, so when you knock out the 2 and the 3, you can still see some calcium events, sparse calcium events. Now, if you do only the knockout of IP3 type 1, my guess is that, I don't know, in fact, you will not see this, the one that is induced for sure involved in LTP induction. It depends on the protocol you're using.
So then maybe you listen to processes and still keep it the strong and the other way around.
Yes, the question is whether the IP3 type 1 activation, is it necessary for the IP3 type 2 then to pick up the button and to be involved in the propagation? And this I cannot answer. That's why the classical, my quick answer will be yes, I'm sure we will see a decrease, but I don't know. You're welcome.
More questions? It's working.
You had a question?
Yes, I had a question. So like the ephrin B3 receptor, right? Is there any way that can be regulated? Like, for example, if you're not good at like remembering things, maybe when you're drunk, like this doesn't work very well, or you didn't look at this?
Why drunk?
Well, I mean, because like, I was just thinking about like when your memory is not working very well. Is this something that is related to the forgetting curve?
I understand your question.
Yes. So that's why I was drunk. I was just thinking of the situation that my memory was not
So
working.
two things. First, what I didn't say, but what we have shown also, is that after the induction, not before, but after the induction, the associative process are retracting, meaning that this binding is not present after the induction. So your question is whether, depending on the situation, is it possible that as the associative process are retracting, this connection is not present? And my answer is potentially yes, I'm almost sure, and this is what is occurring in the hypothalamus. So yes, perhaps when the associative process retracts, this interaction is not there, and therefore the availability in the synapse is decreased. I hope I answer your question.
Yes.
Thank you very much.
Thank you.
Okay, so I guess we are coming to the end. So thank you very much to all of you. I'm waiting for one of the Keynote sponsor, because he told me that there is like sorteo, to do, but I don't see him. So I offer him to do it now that you are all here. You go, okay. So yeah, let's wait just a few minutes if he comes. And then, well, anyhow.